use of Drosophila as model organism to study Mendelian law of inheritance. We are well aware that it was Gregor John Mendel who worked on Pisum sativum, that is garden pea, to explore the way the parental features are inherited to the next generation. Now, same aspect could be studied by using other organisms like Drosophila melanogaster is one of the model organisms which can be reared in laboratory conditions, and we can perform a number of experiments with this model organism. In Banaras Hindu University, Department of Zoology, we have a very good laboratory of Drosophila, and where several stocks of Drosophila melanogaster, its mutant stocks, and several other species of this genus are being cultured. So we have in PG as well as UG classes several experiments related to Drosophila. Now, certain experiments which are related to the genetics aspect are monohybrid and dihybrid crosses. So in this presentation, I am going to explain that how we demonstrate monohybrid and dihybrid cross, explaining the law of dominance, law of segregation, and law of independent assortment. So what we do, we have Drosophila melanogaster stocks, and such stocks are wild type stocks. That is, the flies which are reared in such stocks, they possess all normal features. They will have normal eye color, that is red eye color. They will have long wing, and for every feature they would be normal. And we have nearly a dozen of mutant stocks also. So out of them, one stock is curled winged. So all the males and female flies in that stock will have curled wings. So this is because of a mutation. Now, this curled wing mutation is actually located on the third chromosome, and it is an autosomal mutation. So by using this mutant stock, we can study law of dominance as well as law of segregation. So what we do, we collect virgin female flies from the wild type stock, that is the stock in which every feature of the fly will be normal. We collect female flies from that stock, and then such flies are crossed with mutant male. Okay, so these mutant males, they are used to make the cross. And in the next generation, we observe all the flies showing normal features. It means this normal wing size, so that is a dominant feature over the curled wing. So this is a recessive trait. By doing this cross, if we find in F1 all flies having normal size of their wing, that indicates that this is a dominant feature. Now these F1 flies, they are then self-crossed. It means all males and females which are present in F1, they can be allowed to mate among each other. So if you will do this, you will find in the next generation, that is in F2, the flies will have normal wing, as well as there will be flies with curled wing. But we will find that normal winged flies will always be more in number than the flies which are mutant in their phenotype. So irrespective of sex, we find that there are male and female flies with a normal wing structure, and there are male and female flies with curled wing structure, but their number we expect in 3:1. So, and we quite often find this ratio to be followed. So let us have a very clear idea about this particular cross because this is actually the phenotypic aspect, that is external appearance, that is being considered in this entire experiment. So let us have everything in much detail. What we did, the parental flies we actually used were females, which were normal winged flies, so they could be indicated as Cu plus Cu plus, because we have wild stock where all flies in every generation show this feature, so they have a normal allele, and that allele can be shown by symbol Cu plus. And if only Cu is being written, it is for curled wing. So this is a mutation, this is a mutant allele. Cu is mutant allele and Cu plus is a normal allele. So flies with normal phenotype, normal wing structure crossed with curled wing flies which are male. In next generation, as I just said, all the flies, they will have a normal wing, but genotypically they will be Cu plus and Cu. Now these flies were self-crossed. Males and females with this genotype, they will be self-crossed. So suppose you have a male fly with this genotype. So in that male fly, two types of spermatozoa will be formed. That is 50% of gametes will have Cu plus allele and 50% of the gametes will have Cu. Then in case of female also, exactly the same way, 50% of the eggs will have Cu plus allele and 50% will have Cu. Now there will be a random fertilization between eggs and spermatozoa. As a result of that, Cu plus means if this sperm fertilizes the egg with Cu plus, then you will find this individual. Then there will be individual like Cu and Cu plus, so heterozygotes will be obtained. Here also this is an individual with Cu plus and Cu allele, and this individual will be Cu and Cu. So actually three different kinds of individuals with respect to genotype are being observed. Cu plus, one type, then Cu, this is another type, and then there are heterozygotes with Cu plus Cu and Cu plus Cu. So number wise you can say 1 is to 2 is to 1. This ratio will be obtained in F2. So this is genotypic ratio. Okay, so those individuals who are heterozygous, they will also show normal size of the wing or normal structure of the wing, because this is a dominant feature. So if you add up the heterozygotes with homozygous dominant, then the number will become 3. So the ratio 3 is to 1 will be expected. And here suppose you have performed an experiment. In this table I am showing data of F2. So you actually performed the experiment. You crossed parental flies as I just told. Then you got F1 flies. You allowed F1 flies to mate with each other. And in F2, suppose you get this result. You got normal winged flies in number 1530. And then there were curled winged flies. They were 417 number. If you add up these two, then total comes 2000. It means total 2000 flies were observed in F2 with the number 1530 and 470. Now whether this F2 result follows Mendelian pattern of segregation or law of segregation. So for that one has to apply chi-square test, because through this test you can know whether the observed results follow 3:1 ratio or not. Now for this purpose you know that 3/4 flies will have to be normal winged. So out of 2000, 3/4 will be 1500, because it's 1/4 will be 500. So 500 flies will have to be curled winged, and 1500, it's 3 times more, means 1500 will be normal winged. So you have now expected values, and you have already done the experiment. Observed number is already given. So you have to test whether the difference between observation and expectation, that difference is a significant difference or non-significant difference. So for this purpose, chi-square analysis will be performed. Here I have not written the formula of chi-square, but the formula of chi-square is summation observed minus expected, its whole square divided by expected. So by observing, means with the help of this formula, you can find out the chi-square value for this particular class, that is normal winged class, as well as for the curled winged. It means what you will do, 1530 minus 1500, then the difference will be of 30. So this 30 will be squared. It will be 900, and this 900 will be divided by 1500. So you will get the value 0.60. Then in the other case, 470 minus 500, it will come minus 30. When you will square it, it will come 900, and then this 900 will be divided by 500, and the value will come 1.8. Total will become 2.40. So the chi-square calculated value is now 2.40. Degree of freedom, since two separate two classes are there, so degree of freedom will be equal to number of classes minus 1. So 2 minus 1, that will be 1. And table value will be 3.84. This table value you can see in the probability table for chi-square. At 5% level, the table value is 3.84. So when you compare this table value with the calculated value, that is 2.40, you find that the calculated value is less than the table value. It means the probability will be more than 5%. Okay, the probability of occurrence of this event will be more than 5%, and you thus accept the null hypothesis. And what is null hypothesis? In the initial stage itself, we decide null hypothesis, and that is F2 ratio will follow 3:1 segregation pattern, whereas the alternate hypothesis will be that F2 flies may not be segregating in 3:1 ratio. So since probability of occurrence of this event is more than 5%, we accept the null hypothesis. It means the present result follow Mendelian pattern of inheritance, that is 3:1 ratio. The segregation ratio is being obtained or observed in the present case. So this way you can perform the whole experiment and you can interpret your data. Likewise, we all know that Mendel also performed dihybrid crosses in which two characters were actually considered, and each character was being represented in its two contrasting forms. So you can say two pairs of features were considered. Likewise, in case of Drosophila also, we can consider two separate characters, and those characters could be flies having normal wing, as well as their eye will be red in color. So these are wild type flies which are normal for all in respect to all features. So they have normal wings size and they have red eye color. And then there are mutant flies which have vestigial wing. Okay, this is a vestigial wing, and then sepia eye color. The eye color is also different. So and we have in our laboratory, we have such kind of flies which are actually double mutant. They have mutation in the second chromosome, that is an autosome, and because of mutation in second chromosome, flies have vestigial wing. And then on a different chromosome, on third chromosome, there is a locus for sepia color, and because of mutation in that locus, the flies show sepia color. So double mutants are used. Now these two features, the genes deciding these two characters, are actually located on different chromosome. One is located on second chromosome, that is for vestigial wing, that gene is located on second chromosome, and gene deciding sepia color, that is located on third chromosome. So we expect independent assortment of the characters, or these two genes, they will be independently assorting because they are situated on separate chromosomes. They are not linked to each other. So what one can do, the virgin female flies, which are normal winged and normal eye colored flies, they will be crossed with the male flies having vestigial wing and sepia color. Now in the next generation, you will find all flies having normal wing and normal eye color. So it means these two features, that is normal wing is dominant over vestigial one, and the red eye color or normal eye color, that is dominant over the sepia color. So these F1 flies will then be self-crossed. It means they will be crossed among themselves. So in F2 generation, you will be getting 4 different kind of flies. And these 4 different kinds of flies will be normal winged and normal eyed flies. They will be of one type. Then you will have flies having normal wing and sepia eye color. Then you will have flies having vestigial wing and normal eye color. And the fourth type of flies will have vestigial wing and sepia eye color. And when you will sort out these flies according to their phenotype and also in their phenotype and count them, then you will find that they will be in the ratio of 9:3:3:1. That is, those flies which are normal winged and normal eye colored, they will be 9 in number. Then those with normal wing but sepia color, they will be 3 in number. Those with vestigial wing and normal eye color, they will be in 3 number. And ratio wise, vestigial winged and sepia flies, they will be in the ratio of 1. So this ratio you will be observing in F2. Now we can analyze this entire result, which is actually giving only the phenotypic expression. We can analyze it by seeing it in more detail. What I showed, that parental flies, they possessed normal wing, that is VG plus. VG stands for vestigial wing, and if plus symbol is there, it is for normal wing shape. Then SC plus, that is the red eye color, but SC will indicate sepia color. So VG plus means on both chromosomes, homologous chromosomes, normal alleles are situated. And on a different chromosome, SC plus alleles are there. These flies were crossed with those flies which were mutant male, and they were actually vestigial winged and sepia color, because on their second and third chromosome there were mutations for wing shape as well as for the eye color. Now when they will be crossed in next generation, what will happen? This VG plus and this VG from the other, means male side, will come. So these flies will be VG plus VZ. And for other gene locus, they will be SC plus and SC. So these are heterozygous flies. Genotypically they are VG plus VZ and SC plus SC. And these flies will show normal phenotype. So all F1 flies will have normal phenotype. And if they will be self-crossed, then you will be getting 4 different kinds of flies. That is those with normal phenotype, that is normal wing and normal eye color. Then those which will have normal wing but sepia eye color, or vestigial wing and normal eye color, or vestigial wing and vestigial sepia color. So how that will be possible? What will happen? These F1 flies, in these F1 flies we can see gametogenesis. So there will be male individuals in which gametogenesis will be there. There will be female individuals in which gametogenesis will be there. And 4 different kinds of gametes will be formed in male. Exactly the same way 4 different kinds of gametes will be formed in female. Let us see the gametes. Gamete could be Vg plus and Sc plus. Then Vg plus and Sc. Then there could be gametes like Vg and Sc plus, and there could be gametes like Vc and Sc. That is, on both chromosome the recessive alleles are present. Exactly the same way 4 different kinds of gametes will be formed in female, and then there will be random fertilization among these gametes. So through this Punnett square, in which 16 squares are there. So this is a Punnett square where the fusion of male and female gametes and the result of such fusion means the individuals formed due to fusion of such gametes are shown. So when you will analyze the genotype of these F2, because this is actually showing the F2 result, analyze this one, you will find in this diagram you can see the F2 types will be 4 different types. Actually, the number wise 9:3:3:2 ratio will be obtained if you will see the phenotype. But see here, all those individuals who will be Vg plus and Sc plus, they will show normal phenotype. Likewise, Vg plus Vc and Sc plus Sc, they are heterozygotes. They will show normal phenotypes. Vg plus and Sc plus Sc, they will show normal phenotypes. So you can go through such genotypes, and those who will have Vg plus and Sc, they will actually show sepia color, but their wing will be normal. Likewise, here Vg means vestigial wing will be there, and then the Sc plus means red eye color will be there. And you will have the individuals with vestigial wing and sepia color, so their genotype will be Vg and Sc. So in this table you can see all different kinds of genotypes which will be possible. You can say as a whole 1, 2, 3, 4, 5, 6, 7, 8 and 9. 9 different genotypes will be possible in F2. And 4 different phenotypes will be possible. The ratio will be 9:3:3:1. Suppose, although we demonstrate such experiments to our BSc and MSc students, but suppose you performed an experiment, exactly the same experiment you performed, and what result you obtained, that I'm going to analyze here. That is, you made crosses as I told, and F2 results were obtained. Now you found 920 individuals with normal wing and normal eye color. Then 310 individuals who had normal wing and sepia color. Then 290 individuals who possessed vestigial wing and normal eye color. Then 80 individuals were there which possessed vestigial wing and sepia color. Now total 1600 flies were actually observed. So what we know, that out of 1600, 9/16 will have to be 9/16, because 9:3:3:1. Total becomes 16. So 9/16 will have to be normal for both the features. Then 3/16 and 3/16 should be normal for one feature and mutant for the other feature. And 100 means 1/16 individuals will be of such kind who will have mutation for both the features. So that way you can calculate the expected number. Out of 1600 you got this number: 900, 300, 300 and 100. So you have to now see whether the difference between observed values and expected values come as significant one, or such difference is very negligible, or such difference is insignificant difference. So as I told you in the beginning, you will be performing chi-square test, whose formula is summation O minus E, its square divided by E, where O stands for observed values and E stands for expected values. By doing this, let me tell you, at least for one case, that 920 minus 900, the product will be 20. This will be squared, that is 400, and that 400 will be divided by 900. So this value will come 0.44. Exactly the same way, you will calculate chi-square value for these 4 different classes, and these values come as 0.44, 0.33, 0.33, and 4. Total becomes 5.1. So this is calculated value 5.1. Degree of freedom will be 3 because there are 4 different classes, so 4 minus 1, that is 3. And table value is 7.815, that is at 5% level, table value is 7.815. So you will have to compare this table value with the calculated value, and you find that 5.1 calculated value is less than this table value, so you have to accept the hypothesis, because if you find calculated value more than the table value, then only you will reject the hypothesis. So here the probability is more than 5%. It means null hypothesis is accepted. What is our null hypothesis? Null hypothesis is that the flies which are being observed in F2, they should be in the ratio of 9331, and this you are now accepting, because alternate hypothesis is that F2 flies will not be in the ratio of 9331. Since you are getting this ratio in 9331, you are accepting the null hypothesis. So finally, we shall interpret, or we shall explain, that F2 ratio follows 9:3:3:1 Mendelian dihybrid ratio, and therefore there is independent assortment of characters. So the Mendelian monohybrid and dihybrid, both types of experiments or crosses, could be very well verified by taking the Drosophila flies, their mutants, as well as the wild type flies. If they are being cultured in the laboratory, they can be used to test the Mendelian laws of inheritance.